Hi everyone, welcome to another episode of the Bio-Inorganic Chemistry Lecture Series. Today we are going to discuss dioxygen management in living systems. We know that oxygen
plays a crucial role in
carrying out life processes. We know that in cellular respiration, which is process used for generating energy,
there occur combining of glucose and dioxygen to produce energy along with release of carbon dioxide and water. In other words, we can say that it is a metabolic process that occurs in cells of living organisms by which they utilize molecular oxygen to break down glucose, which is the biological fuel, and produce ATP. Oxygen functions as the final electron acceptor of the electron transport chain, facilitating the movement of electrons down the chain and hence help in producing ATP or the adenosine triphosphate. Oxygen combines with electron and hydrogen ions to form the byproduct water.
the functioning or the proper utility of oxygen can be performed only with the help of dioxygen binding proteins in the living systems. Now, let us see the dioxygen carrying proteins which play an important role in the respiratory processes. The dioxygen carrying proteins include hemoglobin, hemocyanin, hemerythrin, etc. So, these proteins are found in different organisms. In hemoglobin is found in the human beings, hemocyanin in several aquatic organisms, hemerythrin is found in earthworms and certain other lower organisms. So, in each species, the oxygen carrying proteins are different, and in all these things they contribute to the cellular respiration, and these proteins are very important to carry out the life processes. Oxygen carrying proteins are actually metal containing proteins which can efficiently act as biological oxygen carriers for the transport and storage of molecular oxygen. The important dioxygen carrying proteins are hemoglobin, myoglobin.
It is present in
Vertebrates, echinoderms and insect larvae including humans also have hemoglobin and myoglobin. Hemerythrin is found in certain marine invertebrates and annelids like earthworm. We have hemocyanin present in mollusks and certain arthropods. Moving on, now let us see the properties of these dioxygen carrying proteins. We will learn them in detail in the coming lectures. At a glance, you can see the properties of hemoglobin, myoglobin, hemerythrin and hemocyanin here. It is provided that the oxidation state in the deoxy form of the four proteins are provided here. The color of each oxygen binding protein in deoxy form, the color of the proteins in the oxy form is provided here. The coordination of the metal is provided here, the molecular weight and the number of subunits in all the dioxygen carrying proteins are depicted here. We will learn them as the lecture progresses. First, let us learn about the hemocyanin. Actually, hemocyanin is a non-heme oxygen carrying protein. It is found in mollusks and arthropods. It is an asymmetric subunit mass of weighing about 75 kilodaltons and can form chains as large as 460 kilodaltons, which go up to about 6 subunits. The oxygenation process is actually done by a binuclear copper center. The oxygenation process is actually done by a binuclear copper center, which upon oxidation from copper 1 to copper 2 changes the color from clear or colorless to blue. it also function as immunogens. So, the hemocyanin is the oxygen carrying protein in mollusks and arthropods. It is having a subunit mass of 75 kilodalton, and it is usually in these organisms there is occurring up to 6 subunits of the hemocyanin, and the oxygenation process is actually carried out by a binuclear copper center. In other words, we can say hemocyanin is a copper containing oxygen carrying protein. The color upon oxygenation will change from clear to blue color. Here you can see the picture of hemocyanin. The hemocyanin present in arthropods and mollusks are quite different. You can see the shape of molluscan hemocyanin. It can go up to a decamer, that is 10 subunits, while the arthropod hemocyanin is a hexamer. The structure is quite different. You can see the subunits here. You can see the active site of hemocyanin here. It is actually a binuclear copper center, that is the active site of hemocyanin. Now, let us see the structure of hemocyanin in detail. The individual subunits of hemocyanin contains 2 copper atoms which is embedded at the core. The 2 copper atoms or binding sites can be designated as copper A and copper B. They are complexed by 3 histidine residues that form a distorted pyramidal geometry at each atom. You can see here a binuclear copper here. In the deoxy form, that is before binding oxygen, copper is existing in the copper +1 oxidation state. It is a binuclear copper center, that is the active center, and it is connected to 3 histidine residues, and it forms a distorted trigonal pyramidal geometry. So, the geometry here is actually
A distorted pyramidal geometry.
and we have copper +1 at the center.
In hemocyanin, in addition to the 3 histidine residues, there are also 2 phenylalanine residues present in the close proximity. They function as hydrophobic core and protect the active site.
That is, the phenylalanine residues are also playing an important role in hemocyanin oxygenation, and it will protect the active site by maintaining a hydrophobic core. So, you can see the structure of hemocyanin here, binuclear copper center and 3 histidine attached to it with a geometry of pyramidal geometry, the distorted pyramidal geometry. Now, let us see what happens upon oxygenation. Upon oxygenation, copper is oxidized from the +1 oxidation state in the deoxy form to copper 2+ state. Once oxygen molecules bind between the 2 copper atoms, their distance actually changes, and it results in the conformational change of the whole protein unit. Hence, the geometrical shape of the hemocyanin molecule will change from the trigonal pyramidal to the distorted tetrahedral. The binding of dioxygen will distort the coordination of the protein around the histidine that is bound to the copper ions. The 2 copper 2 ions, which is in the D9 form, will get strongly and antiferromagnetically coupled at the room temperature. The dioxygen is reduced to the O22-, which is the peroxide form. We can see that the oxygen is getting reduced to the peroxide form. The evidence was obtained from the resonance Raman spectroscopy, where the oxygen stretch was found to be 803 centimeter inverse, which confirmed the peroxo linkage. Let us discuss the oxygenation on hemocyanin once more. You can see that upon the oxygenation, the copper get oxidized from the +1 oxidation state to the +2 oxidation state. There will be the conformational change occurring within the protein unit, and the geometrical shape will change around the center, that is from the distorted pyramidal, or sorry, the trigonal pyramidal to the distorted tetrahedral geometry. The binding will distort the coordination around the protein, that is the histidine, which is bound to the copper ions. The two copper ions are actually antiferromagnetically coupled at room temperature, and the dioxygen will get reduced to the peroxide form, and it is confirmed by the resonance Raman spectroscopy. So, we can see that in the deoxy form, copper is present in the +1 oxidation state and it is diamagnetic. And in the oxygenated form, that is when it becomes oxyhemocyanin, what happens, the color is being changed from clear to blue and it gets antiferromagnetically coupled, that is the D9 and the O2 will get reduced to the peroxide and it gets antiferromagnetically coupled. Upon oxygenation, the oxidation state will change from +1 to +2 and the color changes from clear to blue, and the diamagnetic nature of the system of hemocyanin will get changed to antiferromagnetic upon oxygenation. Now, we move on to the next oxygen carrying protein, that is heme erythrin. Heme erythrin is also a non-heme iron protein. It is an iron protein, but it is a non-heme iron protein, that is it does not contain any porphyrin ring system. It is found in marine invertebrates like sepinoculates, that is the marine worms, and brachiopods and certain annelids. And heme erythrin and myoheme erythrin, that is the one that is present in the muscles of these organisms, they are actually colorless when deoxygenated, but will turn to violet pink color when it is in the oxygenated state. It is colorless in the deoxygenated form and it changes to violet pink color in the oxygenated state. The heme erythrin contains iron in the +2 oxidation state and it binds oxygen reversibly. After getting oxidized to methemoerythrin, that is Fe3, it loses the affinity to bind further oxygen molecules. That is, after binding or after the Fe2 has getting converted into Fe3, it will lose the ability to further bind oxygen molecule. So, let us see what happens when oxygen is being binded to the heme erythrin. So, this is the heme erythrin molecule. It is colorless in the deoxygenated form. It is containing Fe2 plus ion in the high spin state. So, what happens upon oxygenation? It is getting converted into Fe3 plus low spin and the color changes from colorless to reddish violet. Actually, the heme erythrin exists in the form of an octamer, that is, it contains almost 8 subunits. The active site of the deoxy form comprises of 2 iron atoms. I already mentioned that heme erythrin is an iron protein that does not contain heme, but it contains the iron atom. It comprises 2 iron atoms which are bridged by the carboxylate side chains of a glutamate and aspartate residues. This is the carboxylate of the glutamate and aspartate residues, and
heme erythrin in the deoxy form contain or hold oxygen as hydroperoxide.
You can see heme erythrin holds oxygen as hydroperoxide. So, deoxyheme erythrin has Fe2 plus in high spin state and it is bridged by the hydroxyl group. The hydroxyl group actually serves as a bridging ligand and also it functions as a proton donor to the oxygen substrate. You can see that the hydroxyl group which is present in the deoxyhemerythrin form functions as a bridge ligand and also it acts as a proton donor to the oxygen substrate. It has two functions, it acts as a bridge and also it acts as a proton donor to the oxygen substrate. This proton transfer results in the formation of single oxygen atom, that is a mu-oxo linkage or mu-oxo bridge in oxy and methemerythrin. That is, upon oxygenation, this hydroxyl ligand or the proton transfer will result in the formation of the mu-oxo bridge. The oxygen binds to the pentacoordinate Fe2 plus center at the vacant coordination site. Then the electrons are transferred from the ferrous ion to generate the binuclear ferric center with bound peroxide. So, this is what is occurring in hemerythrin. That is, the oxygen will get binded to the pentacoordinate Fe2 plus center at the vacant coordination site. Then the electrons will get transferred from the ferrous ion to generate the binuclear Fe3 plus center with the bound peroxide. This is the bound peroxide form. In today's lecture, we have learned about
Structure and function of
the oxygen carrying proteins hemocyanin and hemerythrin particularly. We know that hemocyanin is present in certain mollusks and arthropods. It is a copper containing oxygen carrying protein, and the hemerythrin is found in certain earthworm, that is annelids, and certain marine invertebrates. It is a non-heme iron containing protein. Thank you.